You know, deep inside every single one of your cells, there are these tiny little factories working around the clock. They're building the very proteins that, well, make you you. But what if I told you that sometimes these factories deliberately, on purpose, throw a wrench in their own gears and grind everything to a halt? Today we're diving deep into the ribosome's intentional jam.
Yeah, that sounds completely backwards, right? Factories are all about efficiency, speed, getting things done. So why on earth would the fundamental machinery of life decide to just stop? Well, let's unravel this incredible biological mystery together. Okay, so to really get why this factory would ever hit the brakes, we first need to understand how it's supposed to run when everything is going smoothly.
All right, let's meet our factory, the ribosome. The best way to think of it is like a molecular 3D printer. It takes a set of instructions, a blueprint called messenger RNA or mRNA, and it translates that code to build a real functional protein. This is happening in your body trillions of times a second, building literally everything.
And this process is a seriously high-speed assembly line. The ribosome just glides along the mRNA blueprint, reading these little three-letter words called codons. For every single codon it reads, another molecule, a tRNA, zips in and delivers the exact right part, an amino acid. The ribosome then links them all together, and this brand new protein chain snakes its way out through an exit tunnel. It's just this beautiful, perfect picture of molecular efficiency.
But this is where our mystery really kicks off. This super efficient process doesn't always run full steam ahead. Sometimes the ribosome just stops cold, right in the middle of a job, with a protein only half built. And the key thing to get here is, this is not a mistake. This is a programmed, totally intentional stall.
So if this stall is on purpose, who's pulling the strings? Well, in this amazing biological twist, the order to stop comes from the very thing the ribosome is in the middle of building. Let's meet our culprit. This is SecM. As it's being built, a very specific sequence of its amino acids acts like a brake pedal. This little arrest sequence actually reaches out and interacts with the ribosome's own exit tunnel, causing the entire operation to just pause. But why? What's the point?
This is where things get really, really clever. It turns out the instructions for SecM on the mRNA blueprint are immediately followed by the instructions for another protein, SecA. Now, normally the start button for SecA is hidden, tucked away in a little fold in the mRNA. But when the ribosome stalls on SecM, it creates a sort of molecular traffic jam that physically forces that fold to open up, and bam, the start button for SecA is revealed. Suddenly the cell can pump out a ton of SecA, which is a protein that helps other proteins get exported out of the cell. So the stall is actually a sensor, making sure the cell has the right tools ready to go at the exact moment it needs them.
Okay, this feedback loop is brilliant, but for years, scientists couldn't figure out exactly how SecM was pulling this off. To crack the case, they needed to get a look inside the crime scene, to see the machinery right at the moment the jam occurred.
And the big breakthrough came from this incredible technology called cryo-electron microscopy, or cryo-EM for short. What this technique does is flash freeze molecules in motion, basically catching them in the act. It lets scientists build these super high-resolution 3D images of biological machines as they're actually working. It's like having a molecular camera with an impossibly fast shutter speed.
And wow, the images they got were just stunning. We're talking a resolution of 3.3 angstroms. Now, an angstrom is 1/10,000,000th of a meter. That is so incredibly small. At this level of detail, you're not just looking at the shape of the machine, you're literally seeing how individual atoms are pushing and pulling on each other. They could finally see precisely how SecM was physically jamming the ribosome.
But here's where the plot really thickens. As the researchers sorted through hundreds of thousands of these frozen snapshots, they found something they weren't expecting. They didn't just find one way the ribosome were stalled, they found two totally separate populations of ribosomes, each one frozen in a distinctly different stalled position.
And this brings us right to the heart of this entire discovery, the two separate ways that SecM can jam up the protein factory.
So here they are. The first kind of stall happens right before a specific amino acid, proline, is added. In this case, SecM directly messes with the ribosome's tools. The second stall happens right after that proline gets added. And here, SecM doesn't break the tools. It traps the entire assembly line while it's in the middle of moving. Let's dig into each one.
So mechanism number one is basically direct sabotage. Second reaches right into the core of the ribosome, the exact spot where new links are forked in the protein chain, and it physically forces that spot into an inactive shape. This actually blocks the next delivery molecule from dropping off its cargo. The machinery is essentially locked shut. Nothing's getting in.
Now, the second mechanism is a little more subtle, a bit more sneaky. See, after an amino acid is added, the whole ribosome complex has to shift forward to read the next instruction on the blueprint. It's a big mechanical movement. What SecM does here is trap the ribosome halfway through that motion in this kind of twisted, in-between state. This messes up the anchor holding the growing protein, which makes it incredibly hard for the machine to finish its cycle. It's less like breaking the machine and more like, just jamming up the whole conveyor belt.
So let's zoom out for a second. We've got two different ways to stall the ribosome, both controlled by the same little protein. What does this tell us about the bigger picture of how life actually works?
Well, for one, it shows us something profound. Proteins aren't just the passive end products. They can actively reach back and control the very machines that are building them. And this isn't just a simple on-off switch. It's an incredibly sophisticated form of real-time gene control, reshaping the factory's core function with single amino acid precision. This gives us a blueprint for understanding countless other processes in the cell, and even for understanding how some of our most powerful antibiotics work by targeting the ribosome.
I think the researchers who made this discovery summed it up perfectly themselves. It's not just a jam. It's an elegant control system. It's a continuous, dynamic reshaping of one of life's most essential machines, and it's being directed by the very thing it's creating. It's just a beautiful piece of biological engineering.
And so that leaves us with this one kind of mind-blowing thought. If a single protein can hold the instructions not just for its own structure, but for controlling the factory that builds it, what other secret instructions are hiding in plain sight, coded into the fabric of every living thing?